So yeah, thank you for having me. So I'm an associate professor at Washington University in St. Louis. I'm also serving as the deputy director at the National Center for Adaptive Neurotechnologies. And one of the research lines that we're interested there is technologies that can really interact with the nervous system. And what I mean with that is that they can actually use perception or other sensation into the nervous system. But before I start my presentation, I have to disclose my conflict of interest. I hold some intellectual property for brain mapping technologies and may derive licensing income from the same. So let me just introduce the general idea that I want to discuss today. So over the last 20-something years, we've learned a lot about the functional organization of the cortex. Specifically, fMRI studies have discovered a lot of areas throughout the brain that are responsible or are involved in various low-level and high-level function. For example, we know about visual perception, face recognition, we know about motor cortex, and all of this evidence is mostly correlational. Because it was basically observed through indirect means, for example, for an increase of blood flow while the perform a particular task in an fMRI scan. Recently, we have more and more the opportunity to actually observe these changes on the cortex with even more detail and also more directly. The recent advent of electrocorticography allows us to place sensors directly on the surface of the brain or within the brain. These sensors are basically electrodes embedded in a elastic sheet, and they're embedded and placed for clinical reasons. For example, for people that suffer from intractable epilepsy. In these people, we implant these electrodes to localize their seizure onset zone and also to deliver cortical stimulation through these electrodes to map out the brain. And I will talk about that in more detail later. Another interesting aspect about this kind of technology is that it is constantly evolving, that the technology, in this case, the resolution of the ECOG grids is constantly increasing, and we're getting a better understanding of how to actually process these signals. And that's also reflected in the number of applications that is coming out of this field that has been starting off to take off in the last 10 years and has reached over 500 publications per year. But also the quality of this field is increasing, with most of those publications ending up in peer-reviewed journals. So the most seminal discoveries in ECOG is that there are two frequency bands. The low frequency band is very indicative of basically the readiness to execute a particular movement, basically cortical excitation, and the high frequency band that's really indicative of the functional organization underneath. For example, in this case, you see somebody who's performing a motor movement with his right hand, and you see in the left hemisphere hand motor cortex showing power augmentation in the high frequency band and an attenuation basically in the low frequency band that's not so specific and basically makes the entire motor cortex more excitable. We have seen these kind of like patterns, not just for particular single movements. We can do this for various different movements. And we have seen some of that work actually in Christoph Guger's talk just half an hour ago. What I want to get into is that these kind of like power augmentations that we can observe have a very strong correlation with the function that you elicit or inhibit through electrical cortical stimulation. And I will expand a little bit on how to do that actually. So interim summary is basically if you place electrodes on the surface of the brain, we can observe functional changes directly, and we can deliver electrical cortical stimulation and also perturb the nervous system directly, therefore interact with the central nervous system using electrical stimulation. So how do we do that? I want to start out with describing a little bit of the methodology behind that. What we use to stimulate the brain is biphasic stimulation. And these are basically very short pulses. They are 300 microseconds in duration. And the amplitude of these pulses ranges from single milliamps to up to like 15 milliamps. And these pulses are delivered in trains at stimulation rates of 50 hertz or above. And when this stimulation is delivered, what you actually will observe, and I will show this later in examples, is either an excitatory response. For example, if you stimulate motor cortex, you will see the hand move, or if you stimulate areas involved in processing, you will see an interruption of this language processing. There are a couple considerations when we perform this. Obviously, we do this in human subjects, so we don't want to cause any damage or other injury to the cortex. So the safety considerations that we're mostly concerned is that we have to keep the charge density below a certain threshold, in this case, 50 microcoulombs per centimeter squared per phase. So you can already see this is charge density, so this has something to do with the surface area of these electrodes. We can only stimulate electrodes because of that reason that have a certain range of impedance, because otherwise we're not just going to increase the voltage unnecessarily. We have to select what kind of stimulation we perform. There are options that are monopolar or bipolar. These kinds of stimuli, unfortunately, can cause seizures. You've seen this also in Christoph Gruber's talk just half an hour ago. If you stimulate near the epileptogenic onset zone, you may actually start after discharges that can develop into a full-blown seizure. So sometimes you need to either have the anti-epileptic drugs already administered, or you have to have benzodiazepines ready, for example, Ativan for injection if these after discharges develop into a seizure. When we observe the effect on the behavior or the cognitive performance, we typically have to make sure that the stimulation frequency is high enough so that we deliver enough charge to the cortex. So stimulation underneath 20 hertz typically is not very reliable in eliciting a functional response or a change of cognition. The stimulation current has to be within a certain range of safety, and I will illustrate that later. For example, motor cortex can be kindled with 2 to 6 milliamps. So it doesn't take much current intensity to create either a cessation or an actual motor movement. However, language requires much higher intensities and longer trains to be disruptive. Also, the stimulation artifact needs to be visible. You need to make sure that these stimulation trains don't result in interstimulus after discharges, and that they produce a consistent effect. So what that means is that when we perform such a stimulation and we're looking for eliciting a particular effect, we have to perform multiple stimulation trains, and those have to be interleaved with sham trains. And the subject typically is not aware whether you're actually stimulating or whether you're actually performing a sham train. Just to summarize a couple of the safety concerns that we have, obviously the charge transfer. So we have to make sure to stay with below 50 microcoulombs per centimeter square. We have to also make sure that when we stimulate chronically, that the voltage below the threshold for electrolysis, that for chronic stimulation, the thermal deposition doesn't exceed a similar threshold, and also that for longer stimulation, that this doesn't cause any other tissue damage. So, as I said, one of the considerations is how big are those electrodes? You've seen in the very introduction that you can have standard density electrodes. These are the ones that have been used for the last 40 years. They're typically 2.4 millimeters in diameter, and we can have high density electrodes. If we look at the relative surface of these, we can quickly see that high density electrodes are much, much smaller, and therefore the current density will be much higher. So we have issues of how high we can stimulate. The same holds true for depth electrodes, and there another compounding factor is that the fields that we generate are getting much more complicated. So in terms of the safety considerations, as I said, for motor, we just need a few milliamps, but if we need to go into language territory, we need up to like 50 milliamps. So if we look at our 300 microsecond pulse duration and the safe area in which we can operate, we can see that at least for brief stimulation with standard electrodes, we can go all the way up and can elicit disruption of language function. But if we go into high density electrodes, we very quickly go into territory where the stimulus intensity has to be limited. Also want to touch a little bit on the device evolution, because if we actually want to perform this, we have to have devices that are FDA approved and allow us to stimulate or deliver the stimulation safely in humans. So the evolution is kind of interesting because the first standalone cortical stimulators were built and released in the 1970s. This was actually pre-FDA, and they have been around for a very long time, mainly because the market for this kind of stimulation was not very large, and it took until the 1990s until the first company pursued FDA approval, and basically the device pushed out was pretty similar to the device that was in the market before, since the 1970s. And the evolution has been very slow in delivering capable devices. However, recently, electrical stimulation, specifically for the purpose of clinical mapping, has been evolving. For example, this is a system from Neon Coden. Neon Coden provides epilepsy monitoring system. These systems typically consist out of an amplifier system, video system, and it basically allows you to synchronize the signals recorded from the brain with video stream and review them after the fact. They basically provide now a system in which you can click at particular electrodes and deliver these stimuli. This is mainly geared towards clinicians that want to perform stimulation mapping or want to explore the epidemic onset zone in more detail. For scientific investigations, the advent of programmable stimulators has been very significant. This is a stimulator that is now being FDA approved and provided by GTEC. This stimulator can be controlled and is programmable, and what that allows us is to do more systematic exploration of the effect of electrical cortical stimulation, in which we augment the clinician with a program that delivers this stimuli in a more controlled fashion, sometimes in a closed loop fashion. For example, if we want to deliver stimuli at particular phases of an underlying oscillation, with these devices, with these programmable stimulators, we can do that. So I want to show a couple of demonstrations of the effect of electrical cortical stimulation. These are videos that have been generated in the course of being people undergoing temporary placement of these ECOG electrodes and subsequent functional mapping of their cortex. So this first example is now stimulating a human subject with 2 to 3 milliamps in motor cortex.
Oh, whoa, jeez.
And you can see the effect is very strong. It's very difficult for the subject not to notice that its hand is controlled. So again, the subject did not want to control or move his hand. This is involuntarily induced by stimulating hand motor cortex with 3 milliamps at 50 hertz. You can not only, or in addition to making these crude movements, if you have more high density electrodes, you can control the motor function in a more fine detail. So this is an example from Nick Ramsey, University Medical Center in Utrecht, where he basically has implanted a high-density grid over motor cortex, or motor cortex in this case, and he stimulates different pairs of electrodes, okay, that are adjacent, and elicits different directions of tongue movement. So as you can see here in the first example, he is stimulating this pair, which elicits a movement of the tongue to the left. Now he stimulates a pair adjacent to that, which makes the tongue move up. And another example, another pair adjacent to that, now basically the tongue is moving to the right. And these are very reproducible results. And here's basically now all of them superimposed. So we not only are able to induce motor movements, we can also induce perception or disrupt perception. So this is now results presented from a subject implanted at Albany Medical College. This subject had electrodes implanted over fusiform gyrus. This is an area that is involved in recognizing faces. And when stimulating nearby electrodes there, we could observe that color perception was induced and face perception was disrupted. So now I'm going to show you the example for color perception. So the subject is stimulated at these electrodes that I annotated here.
Yeah. Spots.
Where?
Peripheral vision.
What just happened?
There's something blinking.
Where in your peripheral vision?
Top corner. Right.
So you get a very, very specific perceptual
Color.
Effect from the stimulation. More interestingly, now a very adjacent electrodes that's now right on fusiform gyrus. When this is stimulated, the subject is unable to recognize faces.
no longer have any kind of eyeball
No, come on.
on your right side. Right side, I'm not really seeing much.
Now I have the face again.
Yes.
So if you stop the stimulation, the effect immediately stops to persist, and the subject is again able to recognize the face. So interim summary, so what we have learned from these clinical stimulation studies is that it can induce motor movements, motor sensation, perception, and it can disrupt all of these functions as well. Another thing that we've learned is that the effect that we can observe can be distributed throughout the cortex. So you can have the same observation by stimulating a wide range of areas throughout the cortex. Even more troublesome, you can actually, on the same side, depending on stimulation parameters, observe various different effects of your stimulation. For example, you can have numbness and a sensation at the same location depending on the stimulation parameters, or deafness and sounds. So next, I want to touch upon a couple of the clinical utilities that this closed-loop stimulation that you can now perform has. So as I said before, these electrodes allow us to record and stimulate at the same time. So one of the clinical applications is to disrupt epileptic activity that could potentially develop into a full-blown seizure. So what you see here is basically a trace of around 10 seconds where the subject is actually undergoing functional mapping. So the red area on the left shows basically this mapping burst where the cortex is stimulated. Unfortunately for the subject, this mapping develops into what's called after discharges. You can see this activity here on the blue underlaid traces. And obviously, the clinician then attempts to disrupt this ongoing after discharges. So he delivers brief disruptive bursts to the cortex. And he repeats this several times. And on the third disruptive burst, he actually clears these after discharges. So while this is at the bedside, the principle is pretty simple. You observe, you detect that there's something outside of the range of normal activity, you deliver a disruptive burst, you observe. If it's cleared, you're good. If it's not cleared, you repeat. So this principle that has been observed in the clinic for the last 30 years has been recently translated into a device that can be implanted into patients, the so-called responsive neurostimulator. So in this case, you have to first identify the area from where the seizures start. You implant these electrode strips right in clear proximity or above this area. This is fully implantable. It contains a battery that lasts several years, and it operates 24/7. Once it's appropriately programmed, it can detect that the seizure may develop and can try to disrupt it. And this is going to be fairly effective in a variety of different epilepsy conditions. So finally, I want to transition a little bit into some of the ongoing research in our laboratory in which we want to further investigate the scientific utility of electrical stimulation. So in this case, the area of the area in which we are most interested in is single pulse electrical stimulation. So far, I've shown you bursts of 50 hertz. Leave us very little time to observe the effect of the stimulation. However, you can reduce the stimulation frequency to 2 hertz while keeping the same intensity. And this allows us to now observe the functional response to stimulation. However, this is not a trivial endeavor, as the stimulation that we deliver results in stimulation artifacts that are a million times larger than the signals that we observe. For example, we observe signals in the microvolt range. Stimulation is in the volt range. So the requirement to do this is to have an amplifier system that can tolerate these high inputs, and also a technique that allows you to remove these stimulation artifacts and properly perform statistical analysis on the resulting effects. So what you see here in this pipeline is what you need to perform. So you have the stimulation, then you record data from these electrodes, and you can see these huge recorded stimulation artifacts. You have to remove these artifacts carefully without inducing any filter artifacts or artifacts that can result in filter artifacts. Then you can, from that, extract features, in this case, the broadband gamma power after the stimulation in the following 100 milliseconds. You can then visualize the effect of the stimulation, and you can perform statistical analysis across these hundreds of stimulation pulses that you have delivered and determine whether this stimulation results in a functional activation at distant sites. So this is basically our demonstration of this. So in this case, on this subject, we stimulate the tip of the temporal lobe, indicated here by those red dots. And as you can see, this is the functional activation as it traverses throughout the brain. So this is really a short, brief period of time. It goes from the following 20 milliseconds after to less than 200 milliseconds after that. So this is not volume conduction. This is evoked cortical activity in response to this single pulse stimulation. When you do appropriate stimulation analysis, you will see that identify a particular location and the sequence in which they are activated. So thereby, we can do a functional connectivity analysis, allowing us to determine, using single pulse electrical stimulation, how the brain is connected. Here's another study in which we were interested in the effect of areas within this cortical network. So this is stimulating now a subject on the right temporal lobe. So this rectangular feature here is the right superior temporal gyrus. This is the Sylvian fissure. This is kind of like the central sulcus. So we'll stimulate electrodes on the superior temporal gyrus. And as you can see, the entire network within the superior temporal gyrus gets then activated, but not electrodes outside of that. We further investigated that to see whether there's a correlation between the activation that we find in response to single pulse electrical stimulation and that's to natural auditory stimuli. So we have a battery of different auditory stimuli, nature sounds, environmental sounds, and so forth. And we also stimulate the same subject with single pulse electrical stimulation at various sites. So when we perform an analysis of that, this is kind of like the correlation we see. So we see sites that elicit a response to the auditory stimulus, and we see sites that elicit a response to the single electrical stimulation. And if we do this across subjects and across various different stimulation pairs, what we find is a very strong correlation between the responses of these electrodes and the locations. So this tells us that basically we can determine functionally connected areas by stimulating one of these electrodes of this area. Another application that we were interested in is now median nerve stimulation. So this is now not stimulating the brain directly, but the median nerve. In this case, the median nerve innervates a big portion of the hand. And what we were interested in is the somatosensory evoked potentials that are elicited through the median nerve stimulation, or evoked through the median nerve stimulation. And we were interested in two aspects of that: the polarity of these potential, because it has been known for a very long time that they can delineate basically the central sulcus. Basically, whether there's a positive or negative peak indicates whether this is pre or post central sulcus. But also, we speculated that they also induced a functional response, pretty much similar to what I've shown you before. So we looked at the broadband gamma response to this median nerve stimulation. And so what you see here is now a result from one of the subjects that was stimulated that way. So you can see here the central sulcus as determined automatically by the polarity reversal in the evoked potentials. But also, when we do further analysis here, and again, this is like really above, right onto the motor cortex. Okay, when we perform another analysis, just looking at the time frequency analysis of the broadband gamma activity. So you see here from 70 to 240 hertz, the 0 to 20 milliseconds after the median nerve stimulation, we see a not very specific stimulation artifact, but following that, a very specific functional activity of the cortex. And when we perform control analysis in which we perform vibrotactile stimulation, kind of like induce sensation and movement into the innervated area, we can see a very strong correlation between the two, indicating that median nerve stimulation can be used not only to delineate the central sulcus, but also to determine the functionally activated area. So a couple of the conclusions from my talk. So electrical cortical stimulation to directly interact with the nervous system can be delivered through short bursts of electrical stimulation. You can either elicit an effect or inhibit function. You can also perform single pulse electrical stimulation for more scientific endeavors. This has clinical utilities using neurostimulation or scientific utility for human brain mapping. So with that, I would like to give credit to the people in my laboratory and my collaborators, both at my old institution at Auburn Medical College and my I've recently joined the Department of Neurosurgery at Washington University, and also to all these sponsors that provide funding for these endeavors from NIH and the U.S. Army Research Office and Fondazione Neurone. With that, I have concluded my talk, and I'm open for questions. Thank you.